Okay, so hello everyone. I'm Emily Findlay. I'm an associate professor here, and I have been at Southampton since 2022 when I moved my lab here from Edinburgh, where I've been before, but moved to come home because I'm a Portsmouth girl at heart. So I'm going to tell you about the work that I do in my lab a little bit, but give you a taster lecture on immunology. So I'm an immunologist. My lab and where I work with my team actually isn't here in Highfield. We're in the hospital, which is, if you don't know, Southampton is about 1.5 miles away. There's about 4 of us in the biological sciences faculty who work in the hospital. And that's because the sort of biology that we do is really on the cusp of medicine. We are biologists, we're not medical professionals, but the work we do interacts with lots of people in the hospital, so it's easiest for us to be based there. All of our teaching we do here, but we come backwards and forwards. So you can sort of see the work that we do. It means that I'm going to tell you at the end the courses that we teach on, but it means that if you are particularly interested in things like immunology, you can choose to come and do your projects with us in the hospital as well. It doesn't have to be limited to here. Right, so I'm going to tell you about the immune system. So your immune systems, we all know you're all children of COVID era. You're all aware of the importance of an immune system. It's something that your body devotes an enormous amount of resources to. Just huge amounts of the energy of the food you eat, of your body's effort goes into maintaining your immune system. And the reason why is because we are faced with just an extraordinary number of challenges every day. So there was an experiment that was done quite a long time ago now where they get people to breathe out into like tubes with saline in it, and then they count what you have. So if you collect breath from an adult for 1 minute, you will collect on average 10,000 bacteria, 5,000 viruses, and 2 fungi. And that's just what you breathe in or out in 1 minute. If you think again what you touch, everything that we eat, everything that we come in contact with, our body is continually bombarded with challenges that it has to deal with. And actually, we're extremely good at it. If you think how often you get sick, if you're not a parent of a small child in Britain, you probably get sick 2 or 3 times a year, actually. We do an astonishingly good job, considering we're breathing in 10,000 bacteria every minute. So actually, our bodies are very good at this. And we know, the other way that we can know that we're very good at this is we know what happens to people who don't have an immune system. So some of you may know that this is a boy called David. There was a film made of his life with John Travolta in it, so some of you may have seen this. And he was known as the boy in the bubble. So David was born in the 1970s in America, and David had a disease called severe combined immunodeficiency syndrome. And this is a disease which is very rare. It's a mutation in one of 9 genes in the body. And if the babies that are born with one of these mutations, have no immune system. In the past, life expectancy for these babies was less than 1. And the reason for that is that they just could not cope with infection. Without an immune system, they would die very quickly. Nowadays, they're all cured by gene therapy, and they have a normal life expectancy. But in the 1970s, their life expectancy was less than 1. But at the time, in the 1970s, in California where he was, people were beginning to understand, the doctors were beginning to understand that blood transfusion and complete transplant of the immune system could fix this. And so they said, let's keep him in a bubble until that technology is available. And so his parents did this. They kept him in this bubble. He never went outside. He was put in from birth, so he never hugged his mother. And he was kept, all his food was autoclaved, was cleaned, all his water was filtered, and he was in there until he was a teenager. NASA made him a spacesuit so he could walk outside, a special spacesuit. He did, in the end, have a complete bone marrow transplant, a complete immune system transplant, but did unfortunately die a few years later from a tumour that resulted as a result of the bone marrow. But this is what happens when you don't have an immune system. And this is why the immune system is special in your body. It takes up to 30% of your metabolism, your basal metabolic rate, about 30% of it is used to maintain your immune system. And that's why people who don't eat, perhaps at the end of life, don't take in enough calories and get lots of infections. Because about 30% of your metabolism is just to maintain the enormous amount of energy that your immune system needs. Immune cells are special. They can go anywhere. So normally in your body, cells stay in one place. Your liver cells stay in your liver. Your muscle cells stay in your muscle. Immune cells can go anywhere. They can travel in and out. They can travel through your liver and then go through your intestine and then go to your blood. And that's because they have to see everything that's happening throughout your body. Some of them can survive an extraordinarily long time. So recently you might have heard about the monkeypox outbreaks that have been happening. People who were vaccinated against smallpox in the 1970s were protected against that. And that's because the immune cells that were induced by their vaccination against smallpox protected against mpox, as we now call it, when mpox came back. So 50 years later, that vaccination still held true. And so the immune system, just keep an eye on the time, is very, very important. It's also unbelievably complex. Now, if any of you are thinking of coming here, don't worry, I would never ask anyone to understand this. I'm just showing you that this is a tiny part of the immune system in one cell. It is extraordinarily complex. People like me have devoted my life to understanding maybe one of these little points. And this is what we do all day long. And the reason it has to be complex is that as well as facing a huge number of challenges, we face a huge complexity of challenges. So every day, for example, every time you touch some food or eat some food, you will encounter bacteria. These are very different. Some bacteria live in your lungs, some live in your intestine. The ones that can survive your stomach acid are very difficult to kill, and they're all slightly different. Some of challenges we face are airborne, as we all know now, like COVID or like the influenza virus. Viruses, in particular, mutate incredibly quickly, as again, you all know, because now the whole country is full of immunologists. We were all immune to the alpha variant of COVID, right? And then suddenly it mutated and Omicron came and none of us had any immunity again. That's what happens with viruses. They mutate extremely quickly. The route that infections get into our body changes what sort of immune system we need. So as an example, if you eat, in Britain, the main culprit for food poisoning is bags of salad. So if you eat a bag of salad and get E. coli poisoning from that, those E. coli have survived your stomach acid and got to your intestine. Now, the immune system that you need to deal with that will be different than if you're bitten by an insect and are infected with malaria. You need an immune system there that's in your skin. The malaria parasite then moves to live in your red blood cell. You need an immune system that can cope in your blood, in your spleen, in your liver with these parasites, which is very different to the immune system you need throughout your nose and throat if you've been infected with flu. And so the size of the challenge as well, if you think that the immune system has to cope simultaneously with a malaria parasite, which is so tiny it lives inside a red blood cell, and a tapeworm, the record length of which is 25 metres. The idea that the same immune system has to cope with both means that we have just an extraordinarily complex system we need to develop. Now, what I'm going to tell you about for the last 15 minutes of this is not actually about infections. So the other thing that the immune system does, which is extremely important and which Southampton is unbelievably good at researching, is how the immune system kills cancer cells. So the immune system doesn't only kill infections, it also kills cancer. And again, this will happen in all of us every day. Probably every day we have a cell that becomes cancerous in our body and it's killed off by the immune system and it never develops. Now this is the sort of experiment and the sort of graph that I would show you. Actually, I show this to my final year students, so I'm going to explain it to you today, but this is the sort of thing I might show in the final year course when we learn about the immunology of cancer. And what this is this was an experiment that was done, and it's what's called a meta-analysis, to understand whether the immune system kills tumours and which ones they might kill. And in a meta-analysis, you go back through all the medical records of millions of people and see the differences between them. So these are people who all have kidney disease, and they were all given a kidney transplant. Now, when you're given a transplant, you may know you get given very powerful drugs that switch off your immune system. And the reason for that is you're given someone else's kidney, and your immune system would normally try to kill it. So for the rest of your life, you're on immunosuppressant drugs. And they're the transplant group, which are in blue. Now, for some of these people, that transplant failed. And so they were then taken off the immunosuppressant drugs. Their immune system came back, and they went back onto dialysis. And so if you know from your A-level studies that every experiment needs a perfect control. This is the perfect control group. because these people have all got the same disease, they've all had the same treatments, but at one point some remained on immunosuppressant drugs without an immune system and some were taken off that. And then we can say, if we look back through the medical records of these millions of people, what happens in terms of their cancer? And you can see, if you look at the blue, everything to the right of this line means they get more cancer than the control group, and this line means they get less. So you can see at the top here, these people who had no immune system because they were on kidney transplant, have, oh, the graph's cut off at the bottom, they have their rate of getting Kaposi's sarcoma, which is a type of tumour, is about 100 times increased. And we now know this is linked to infection. If anybody has seen Philadelphia with Tom Hanks, that's what Tom Hanks has in it. So this is hugely increased. Again, also non-Hodgkin's lymphoma increased in people who don't have an immune system. Now, some of the cancers weren't. So stomach cancer, for example, there was no difference between these groups. So by doing this sort of analysis, and this is the sort of thing that you get to learn how to understand and maybe design your own studies in your final year, we can say these types of cancers are ordinarily killed by the immune system. And in the people who are on dialysis, they're being killed. These people are not getting these cancers, but in the people who don't have an immune system, they are. So what type of cell in your body is killing these cancer cells? Well, they're called T cells. They're called T cells because they're made in the thymus. And they're my favourite cells. These are in blue. And they're produced, you have millions of them in your blood. If you bleed someone, and we do in my lab, we bleed people every day for experiments, you have about 1 million of these in each mil of your blood. So you have huge amounts of them. And these are the cells that kill your tumour cells. And how do we know this? We know this from, again, the same sort of studies. And this is exactly the sort of study that we do in Southampton a lot. There's a whole centre of cancer immunology in the hospital. And again, you can do your projects on it if you're interested. These are biopsies of melanomas, of people with melanoma. And they've stained them in the lab, and each one of these purple dots is a T cell. Now, what they've done here in this study, and this was actually done in Australia, which for obvious reasons has just phenomenal research into melanoma. What they've done here is they've got just thousands and thousands of melanoma biopsies when they've been removed from people. And then they stained them and looked at all the T cells, and then nowadays we do this with AI, but this was done 20 years ago, so they had to get some poor people to click every time they saw a purple cell. And they said, let's group them into 3 groups. Let's say some people's melanoma, you can see here, has no T cells in it. It's all pink. Some people have a few, some have lots, and some have an enormous amount. And then we can say, are the outcomes of these groups different? Bearing in mind that these people are all in Australia, so they've got one of the world's best medical treatments. They've all had surgery to remove their melanoma, and they're all on drugs. So even all of that, is there a difference between the outcome of the cancer between this group and this group? Mm-hmm. And you can see that there is. So this is survival in months. And what this is saying, the red group here are these people, and the black group are these people. So what they're saying is, if you have T cells that go into your tumour, that is the single factor in all of treatment that determines that you're going to survive for up to, well, this is past 160 months. That's when they stop monitoring. So they survive for a very long time, more than 10 years. So having T cells move into your tumour is the one factor that makes an enormous difference. And why is it? It's because T cells can kill cancer cells immediately. So this is a video of one that I just got off Twitter, but it's real. It's from a lab in the States. This pink is a T cell. This green thing here is a cancer cell, and it's going to release some red killer mediators from the tumour. Let's have a look. So you can see, there goes the red, and then there's the cancer cell dying. So that's what they do. I can check the time. Right. So the question is, why doesn't this happen all the time? Why do we get cancer? If you've got 1 million T cells in your blood, per mil of blood, and they can kill tumours like that, why do we get cancers? And the answer is, tumours evolve to hide from the T cells. And tumours mutate and evolve very quickly. And there's a whole module you can do in your 3rd year on cancer biology, which will tell you all about this if you're interested. If T cells can be made to see tumours, they can often kill it. but the tumours are very good at hiding, and particularly if they're in a place where you wouldn't notice, and that's why people tend to survive more from skin cancer than from something like pancreatic, where it's very hidden, where we can't see. And so there's a couple of therapies now that people are doing which are really exciting, where they just say, okay, T cells are good at seeing some things like viruses. What if we put a virus into the tumour? And unbelievably, this works. So this was done in the States. One of the hardest cancers to treat is brain cancer called glioblastoma, which is it's not a single tumour, so it can't really be cut out. But what they did there was they just injected the polio vaccine into the tumour in the brain. And the polio vaccine has poliovirus in it, and that just alerted the T cells to the immune response. And whereas these patients almost all die within 5 years, 6 years later, 20% of the ones that got this treatment were alive. Because all it did was it just made the T cells say, oh, right, there's a tumour there, whereas normally it had been hidden. There was also a great story from just over a year ago about a virologist, I think she was in Poland, who had breast cancer for the second time. She knew it had come back, and she was growing viruses in her lab. So she just injected her tumour with the viruses and cured herself. And then got sacked for huge breaches of health and safety protocol. But she survived. So this was again, if you can tell your T cells that there's a tumour here, they're much able to deal with it. So the next thing people asked, and this was about 30 years ago, was okay, can we therefore switch all the T cells on in our body for someone who's got cancer? Will that go and kill it? Because one of the other things that tumours do is not only do they hide from tumour T cells, if T cells get in, they can switch them off. Tumours are very good at doing that. So they said, okay, we're going to develop this thing. It's called checkpoint blockade, whereas normally a cancer switches off a T cell. We're going to stop that happening with some drugs. And so they did this. They developed this. This was done all over the world, and it is astonishingly good. So this is slightly old, this data. It's even better now. For patients with melanoma who get a single dose of this drug, 25%, their cancer goes away and never comes back. 72% are still alive at 3 years without the need of any further therapy. This, again, some cancers work much better than others with this. So melanoma is particularly good, but it's not any good at things like pancreatic, unfortunately. But even so, it's great. And this was awarded the Nobel Prize for Medicine in 2018. Jim Allison, who's in the States, and Tasuku Honjo from Japan shared the prize for this, for development of these treatments. So every year when I talk about this, students say, why don't we all switch our T cells on all the time? And then we wouldn't get cancers developing. And this is an extremely good question. It's a very logical thing to ask. Unfortunately, the reason we don't do that is that T cells are unbelievably powerful. You all saw the video of it killing that cancer cell immediately. The trouble we have is that sometimes they just direct it a bit wrong. So if, for example, you've got a tumour in your lung and your T cells come along and they can kill that one cell that's tumour, they'll probably also kill the 3 cells either side of it. Or if you've got, like we all do all the time, you've breathed in a virus, you've got a virus infection in your nose, your T cells will come along and kill the cell that's got a virus in it, but they'll also kill a few cells around it. And if you do that on a large scale, you get a lot of damage to your own body. But also the fact that if T cells get into the wrong parts of our body, they can cause huge damage. So, for example, the brain really can't cope with any T cells being in there. The structures are too delicate. The same is true for a fetus and a pregnant woman. If T cells get in there, that it's just too delicate. Or the eyes, for example. And so what happens, that's what we call, if T cells get very switched on, and they go into the wrong place, and they start killing our own tissue, we get autoimmune diseases. And all of the autoimmune diseases on here are all caused by T cells that are too switched on. So ulcerative colitis and Crohn's disease is caused by T cells in your bowel that just produce too much, and they start killing the wrong things, too much of that red that you saw being released. Type 1 diabetes is caused by T cells in the pancreas. Multiple sclerosis in the brain, psoriasis in the skin, rheumatoid arthritis in the joints. All of these are caused by your T cells. And so we have to keep it perfectly balanced. And this is what my lab works on. It's what many labs around the world work on. What can we do so that we can just activate our T cells enough so that they kill the cancer cells and they kill off our COVID that we've just breathed in, but they don't then cause autoimmune disease. And in fact, that drug that I told you that we now give that treats melanoma very well, almost all the patients who take it, melanoma is cured, but they almost all get autoimmune disease afterwards because their T cells have all been switched on. So here, this is another sort of graph that you might see in our studies. Blue is relatively the number of patients who've had this drug who get these diseases, and red is the severity that they report. So you can see, for example, that probably every single patient who's had this drug has skin inflammation afterwards, folliculitis, something like that. And all of them, unfortunately, get nerve pain. And you can see that the red is, for the nerve pain means that's probably extremely painful. And in fact, we know now there's almost a direct correlation that the people who get the worst side effects have the best outcome, unfortunately. So what can we do next? This was all drugs that were developed in the 1990s or the 2000s. What are we working on now? Well, this is what everyone's doing around the world. T cells are all different. There's lots of different types. So we can say, can we find just one little group of them that have a unique role? So there's only a few that kill the cancer cells, and we can only activate them and leave all the rest. That's something that happens. Can we change them so that these drugs only work in the tumour and then can't go anywhere else in the body? Or can we do combinations so that we can switch them off afterwards? Or even things like very carefully changing the dose so you can activate T cells only for a week and then switch it off so that they don't all then go into the intestine and change that afterwards. So that's just an example of the sort of immunology that we do here at Southampton and that you can learn about. I've just finished by telling you about the courses that you can learn it on. So I've just last week been doing the Introduction to Immunology in the first year, which is in the core module that everyone has to do, Fundamentals of Cell Biology. In the second year, I run this course, Immunology, Infection and Inflammation, which is compulsory for biomedical science students, but everyone can take it, so it's optional for everyone else. In year 3, there's another immunology course, which is run by the Faculty of Medicine, but all biology students can take it, which is very good, advanced course. There's also cancer biology in the third year, where we talk a lot about immunotherapeutics, like I've just been talking about. And there's also biomedical technology, where they talk a lot about how they design that sort of therapy. And then in year 4, like I say, if you do a fourth year, you can do a research project with me or with anyone else in the lab. And this is a picture of my student from last year, Carla. This is in my lab in the hospital. This piece of equipment is about £500,000. You can see I was there just to check she was all right and took a photo, and then I left her because she's been well trained on it. She was properly trained. There's lots of support, but she was running these experiments on her own. This is with patient samples. Doing all the work on her own, got a great dissertation. She's gone home to Spain and got a job in a research institute in Spain, so she's done very well out of it. I think that's all I've got to say to you today, but please do ask any questions you would like about immunology or about the course, and I will answer any that I can. But I'm just going to switch off the recording so that none of your questions are recorded on here.